Okay, thanks very much. And also thanks for allowing me to present this work over Zoom. So today we're going to be actually doing a hands-on workshop, but I thought I'd start off with giving a little bit of an introduction, a few slides just to set the scene about what I want to show in the next hour and a half. Now, I will say from the outset, I've no idea how long this is going to take. I haven't rehearsed the timing on this, so I may take much less time. I hopefully won't take much more time than has been allotted, but if I finish early, apologies, you have been warned. What we're going to be trying to do today is using polymapR and polyQTL to do a couple of things. And we're going to be focusing on triploidy, so a triploid population. We're also going to be looking at translocation in that population. And finally, we're going to be using polyQTL to screen for non-additive effect QTLs. Now, to get all these attributes into a data set and a nice compact data set for today's workshop, I had to simulate a data set. So I'm not actually working with a real data set, but I've added a number of, shall we say, realistic attributes. But you will see it's quite an easy analysis. But I hope for some of you working in some of these types of, with some of these issues, that what I'll be showing will provide some inspiration and some help. So just to kick off, triploidy. It's a condition that's in some cases exploited by breeders. In some cases, it just exists in some of our food crops. For instance, in certain fruit crops, seedlessness is a very desirable trait and can be attributed in some crops to triploidy. So it's a ploidy manipulation that's often used for particular purposes. Also in ornamentals, for various reasons, either to make a wide cross between different ploidy levels in a species, or to, for instance, reduce the pollen production of a particular ornamental cut flower. Triploidy or cross-ploidy populations or cultivars have also been used or are being used. And one of the avenues for triploid production is indeed to cross a diploid and a tetraploid. I'm not talking now about, shall we say, more naturally occurring triploids, but if for a breeder would want to deliberately make a cross between diploids or tetraploids and release a triploid cultivar, if you want to do mapping in that, well, then you obviously need to have some software available to do that. So polymapR has been set up to also map triploid populations. It's not something I've shown here at the SCRI sessions in the past, but I thought it would be nice to finish off for this year's workshop to look at the capabilities of cross-ploidy populations in polymapR. What I've done also with this update is I've also simplified the pipeline itself. In the past, if you wanted to map in a triploid, you would have had to have chosen the maternal parent to be tetraploid. Now that's been, shall we say, that demand is no longer there. And as well, the pipeline is a bit more simplified, so you don't have to do all these various tricks that you had to do in the past. So it's more in line with the rest of the ploidies handled by polymapR. And essentially, it relies on mapping at both the diploid and the tetraploid level at the same time, or in your two different parents. And we'll see in the workshop that the diploid pipeline, the diploid mapping approach is slightly different to the one that's usually presented or has been presented so for polyploids. And one of the things there is the two phases between markers, so coupling and repulsion phase, are equally informative. So we have to do something different when we want to do phase assignment. Essentially, it's actually an easier job. We get phase assignment directly from our recombination frequency estimates. Okay, so I'm going to be going through this, as I said, in a couple of minutes. The next thing after triploidy is translocations. As you know, translocations are large-scale chromosomal rearrangements. So that's where part of the chromosome breaks off and it joins to another chromosome sometime in the past. But we can see this if we start to do synteny studies across species. And you can see here this lovely recently published paper across different fish, jellyfish, and so on. You see a beautiful plot of the relationships and the conserved synteny across these chromosomes, but you also see little parts of some of these chromosomes have been, let's see, have been joining into other chromosomes and so on. So when you start comparing across, particularly across species, you can find evidence for translocations, or in some cases even reciprocal translocations. And one very famous reciprocal translocation, coincidentally in a triploid species, or at least in banana, is one that has been well described in the literature between chromosomes 1 and 4. I mean, there are a number of other translocations in banana, but this is the one that's quite prevalent in a lot of the breeding germplasm. And see, from this previous publication from Martin et al., you can see a various number of effects going on here. So these heat map colors here correspond to linkage estimates. So you can see here there's evidence of linkage across these two chromosomes. What you see in the black line, I think, is segregation distortion. So you get high segregation distortion around these regions. And also in these red track over here, it's the recombination frequency. So you can see a suppression of recombination as well around these areas. Now, this was a particular example in a previous study. I was also involved a number of years ago in a mapping study in banana. And there we found also evidence for this particular translocation between chromosomes 1 and 4. But in our study, we were lucky enough that the parents were homozygous for that translocation. So it actually mapped beautifully. So we didn't have this issue with cross-chromosomal linkage, at least in terms of cluttering up or making a mess of linkage estimates between linkage groups 1 and 4. They were very clean. But you could see that when we compared it to the reference genome, that this translocation popped out very nicely. So this is the basis of the data set that we're going to be working on today. As I said, it's simulated, but it's using this as a basis for this example that we're going to be doing. Now, there are some consequences for mapping of translocations. I'm just going to go through them a little bit quickly. There's 3 that spring to my mind at least. And the first of which is a pretty simple one. If you use the chromosomal assignment, so the known chromosome position of your markers on the reference genome, to assign your markers to linkage groups, well, things are going to go wrong if you have a translocation. So assuming here we've 2 collections of markers and we think we know where they should be, the ones colored sort of beige, we assume are on chromosome 1, and the ones that are colored green, we assume are coming from chromosome 2. But if there's a translocation, so part of that chromosome has actually moved across and joined and fused, some of these markers that may actually be linked with each other in our population and actually be part of this other chromosome, but we don't know that in advance. So if we use reference genome to assign our markers to linkage groups, that's going to start creating problems. And we may not actually be aware of why these problems are happening. Of course, there's a very simple solution to this, and that's just to use genetic linkage to do your clustering and linkage group assignment. So, of course, in a way, it's a bit of an artificial problem if you're just using a reference-based clustering. But I see, as also in the previous talk, the genome-based grouping was used, which is fine. But if there are issues that your reference genome isn't consistent with your particular material, it could cause issues. The other consequence for mapping of a translocation is if your parents are heterozygous for a translocation. Now, this is going to lead to a lot of issues. I mean, we saw in one of the previous slides the issue of segregation distortion, or suppression of recombination, but you can also get linkages across chromosomes, which will make it very difficult for mapping. Now, for this particular workshop, I'm not going to be dealing with this issue. It's a little bit challenging. So I'll just leave it there and say, if you happen to be, unfortunately, in a situation where you have parents that are heterozygous for a translocation, well, then there will be fertility issues probably with those parents as well. So that will show up, I think, in your population as well. There is one other possibility regarding translocations, and that's if your parents differ regarding their constitution for a translocation. The main issue probably there is the integration across parental maps is going to be difficult. So if you imagine again a situation where we have 2 parents again, we're just looking at a number of linkage groups or chromosomes. And in parent one, if you can say this is the reference chromosomes as such, whereas parent two is carrying, in this case, reciprocal translocation between those two chromosomes. Now, if this is your situation, so if you've got a cross between parents that are like this, when it comes to integrating those maps, usually we assume that our parents are contiguous in terms of their gene order along these chromosomes, but you're going to have a big issue in these areas where you have that translocation. Now, the obvious thing to do in this situation is to separate out your parental maps. So just produce separate maps for the male and female parents. Again, with polymapR, this is completely feasible to do. There's no difficulty in it. In fact, I think it's even shown how to do this in the vignette. But in any case, we calculate our linkage information per parent anyway. So this is no work as such. But the problem with it is if we want to further down the line do an integrated QTL analysis, we won't be able to do that because we have all these extra linkage groups that are parent specific. Now, there could be another way around it, which is essentially to split up your maps. So in that case, you sort of undo the work of biology and try to correct for this translocation. So you could, in a sense, correct this by chopping them up and then integrating the parts that you know are contiguous or synchronous as such across parents. But that again leaves you with an issue that you have all these shorter chromosomes and not the full length chromosomes. Again, this is not something that you may wish. You could go for a combined approach. So you could use, for instance, a parent-specific map like we had earlier, and you could use the linkage information within that parent to bridge that gap. And so either artificially, you could figure out what's the genetic distance between those ends, and you could try to figure out the relative orientation of those. Or you could use a tool. I mean, in the past, we used to use LP-merge a lot. LP-merge would very easily be adaptable to this particular application. But for subsequent steps then, for QTL analysis, you're going to run into difficulties again if you use this sort of artificially integrated map. Okay, as I said, in our workshop today, I'm not going to be dealing with these types of situations. I'm going to be using the situation that we came across previously in our mapping project in Banana, in which case the parents were homozygous and both shared the same translocation. So it was it was less of an issue. The third topic that I want to cover is non-additive QTLs. I mean, in polyQTLer, we use a basic additive effects model for our QTL scan. So here we're associating again these what we call IBD probabilities, but actually more accurately called posterior haplotype probabilities of inheritance of particular parental homologs. And these xi in this equation here range in between 0 and 1, so they're probabilities in the sense when bivalent pairing has been assumed. They can also, in the case of double reduction, assume a value of up to 2. Now, in the past, we found this is quite a robust method to detect QTLs, but it was brought to my attention that if you have a QTL which has absolutely no additive effects and yet has allelic interaction effects, that you won't be able to detect it with this model, which is true, but most of the time this model will capture also dominant QTLs. I mean, it's quite difficult for you to construct, at least from a simulation point of view, a situation where you only have interaction effects but no main effects of the alleles themselves. But it is possible. So if you want to visualize this here on the left, we see a sort of a distribution of phenotypes across the population. And in this basic additive effects model, we're just trying to associate our phenotype with the presence or absence of particular parental homologs. Now, what we actually get in our when we're estimating these probabilities using hidden Markov models is actually genotype probabilities. So we actually have the probabilities of combinations of particular parental alleles rather than per se these haplotype probabilities. And this approach was actually already proposed by Christine Hackett way back in 2014, is what she calls the complete model, which is instead of just using your haplotype probabilities, just to use your genotype probabilities for QTL detection. The issue, I would say, with this approach is that it starts to become a very large model, particularly at higher ploid levels. So the number of possible combinations that you start to get, particularly if you allow for multivalence, becomes very large. So at the triploid level, which is what we're going to be looking at today, for the bivalence only model, there will be 12 regressors in that model, allowing for multivalence that increases up to 36. But you can see, for instance, if you're working in a hexaploid, where you also allow for multivalence, and again, this is an option in polyQTLR, yeah, the number of parameters that would be needed to be estimated in that model become astronomical. So I'm certainly not motivating that a user should only use this particular approach for a QTL scan, but I thought, yeah, it's a relatively easy extension and may offer some extra abilities or insights to detect to detect other QTL if they happen to be completely non-additive. Let's put it that way. So yeah, again, to visualize this, so we're making an association between our trait distribution on the one hand, and this the regressors here now are the combinations of parental alleles rather than just the probabilities of the parental alleles themselves. So here you see ultimately what we would like to do to come to is a comparison of results using the additive effects model and the full model. So this is something that we're going to be hopefully producing today in today's workshop. Just briefly acknowledge, of course, Chris and Roland, who've worked together with me on this over the past few years. And also I should acknowledge Herman, who originally asked me whether polyQTL would be able to do this or not. He motivated he inspired me at least to add this extra bit to the package. So today's workshop materials are actually on the website of polyploids.org. You're 3 clicks away from having everything you need for today's workshop. You have to go to the workshop banner on the banner, the menu itself, I suppose, the drop-down menu. From there, you just click for the 2024 workshop. And from there, if you go down to my particular talk, you'll see a link embedded there, thanks to Delaney for doing that. So we're going to swap over now. Just 2 minutes. So far, so good. You can hear me okay? Okay. Now, I hope people are willing to follow these steps with me. I mean, I can just show you, but I'm hoping that you'll also be able to actually go through these steps with me. Okay, so as I said, we go to the workshop. So we go to 2024, and we go here, and you'll come to a Google Drive folder. I'm actually going to just download it. So yeah, if there's issues with the data, then this is live. I will find them out as well. But I'm doing this deliberately as well because I want people to have a bit of time to do this. So I'm going to open the folder. Okay. Apparently the internet is slow in the Netherlands. And I'm going to extract all those files. And I'm going to just put them onto the desktop for now. Here we go. And I'm going to extract them all to the desktop. So it's going to make a folder for me now, all going well on the desktop. Okay. And there it is. At the same time, I've launched RStudio, and I'm going to create a new project. Don't worry about that. Okay, so Maybe I should zoom in here a bit. Okay, so I'm going to choose existing directory, and again, I'm going to go to my desktop, and I'm going to associate a new project with that folder. Now, just point out within that folder as well, and that's the let's see. Okay, so within this folder, you also have that PowerPoint I just showed, and there's also a sort of a vignette for today's workshop called triploidmapping.html. So you can open that up as well if you want, and it goes through all these steps in a bit more detail. But for now, I'm going to swap over to RStudio. I don't mind about the updates. And I'm going to open up that R script. So it's called triploidmapping.or. Okay. Now, unfortunately, I'm not in the room. Usually at this stage, I start to walk around and look whether people are on Facebook or whether they're actually with me. But okay, I presume some of you at least are at this stage. And if not, well, you'll have the YouTube video that you can always look back on if you want to in the future. There have been some updates to polymapR and to polyqtl. So in order to get those updates, well, we'll install the package again from CRAN. Yeah, so the current version is 1.1.5, so that's correct. And then we'll just load it into the library. The dosages, so the genotype data, should I say, that we're going to be using is called triploid genotypes. It's in a CSV format. As I said, it is a simulated data set, unfortunately, or fortunately, for whatever, whether you're a purist or not. But it does, yeah, it was simulated using Pedigree Sim, and then I added some particular features to make it a little bit realistic. So it's a data set with about 4,000 markers and about 180 or so individuals. And you can see already in the previous call, I've specified the row names to be column 1, so it should be read in like this. So it's got a certain proportion of missing values. And you'll see already the marker names are informative for the chromosome and base pair position of those markers on that reference banana sequence. Okay. So we're going to do a couple of steps first that we often do in preparation. We start with filtering the data set, so using the checkf1 function. So this is, again, a function that was developed by Roulant, who should be there among you. It's part of the FitPolyTools package, but it's also, shall we say, under the hood in polymapR because it's such a useful function. Now, in this case, we're providing the polysomic argument to be true and setting disomic and mixed to be false. I'm not going to go through all the particular options for checkf1. If you're interested, it's a lovely function. You can get the documentation for that function there. But we're just going to right now run it well with the usual arguments. In this case, the ploidy of our population is, as I said, it's a cross-ploidy population. So the ploidy of parent 1 is diploid, and the ploidy of parent 2 is tetraploid. So they have to be specified. In the past, if you're just using, for instance, a normal tetraploid population, you would just use the argument ploidy, but ploidy 2 is there as an option if it's not the same as ploidy 1, so the ploidy of parent 1. Now, the output of the polymapR version of checkf1 is a list. which is slightly different to the FitPolyTools version. And it has a number of objects within that list. And the one that we're interested in is the checked F1 object. So you see this is a object of 4,312 rows, so the same dimensions as the number of markers in the data set, and has 24 columns. And those columns goes through quite a few different aspects. So it gives the marker number, the name, it gives the consensus parental scores. It also gives the number of offspring carrying, in this case, a dosage of 0, 1, 2, 3, and the number of offspring with missing values. So it produces a nice summary of your data. And it also gives predictions on what's the best model that fits that segregation in that offspring. And again, if you want to look back at, I think, the 2021 SCRI video recording of Roland from FitPoly, I think he went through the check F1 function in much more detail. What we usually look at when we're looking at the output of check F1 is these quality scores. There are two sort of summary quality scores, one which is just simply the product of these three quality factors, and the other one which uses a weighted sum of those three quality attributes. But something that I realized a little bit later when I was using it is also important to check whether your consensus scores, parent 1 and parent 2, are actually scored or not. So for instance, when I say scored or not, whether they're given a value or not, because the function will often return missing values in these columns, and that's actually quite important. Now, in this data set, it's a very nice data set, so there's only one marker where both parental scores are missing. And when I say parental scores, I mean the consensus, the predicted parental scores by check F1. Now, in this particular marker, it's been fit as a 1 to 2 to 1 segregating marker. If we compare that with what we see, so the observed counts of offspring with those particular scores in the population, we see a somewhat significant flag from a chi-square test. So we've got a somewhat significant p-value there. So basically, check F1 is telling us that our parental scores and our offspring scores aren't really matching well, that there is some issue with this marker for whatever reason. Now, of course, in this data set, there's only a single marker. It is slightly unexpected, the segregation, but it's not enough for me to filter this single marker out. I'm not going to bother with that. But I have seen in other data sets where you get a lot more markers which have issues with their segregation and are flagged by check F1. But they curiously, sometimes markers which have an issue and where the consensus parental scores aren't given will still have a relatively high quality score. Because I mean, if we look also at this distribution of our quality score here, this is the Q-all weights distribution. You can see in this case, almost all the markers in this data set have a quality score of close to 1. But I have seen instances of markers which have quite a high quality score, but at the same time, their consensus parental scores are being flagged as problematic by the function. So it's certainly worth your while also to check these and to go into a little bit more detail about what's going on with these markers and why are you flagging this. And I actually, it's very nice to see that after me, I think David Jarrett will be talking also about issues with segregation in populations, in F1 populations, and comparing expected segregations. So that's quite nice to see as well. Okay, I'm going to move on from that, and the next step that we often do is we run a PCA on the population. Again, we're using just the pure dosages of the population to see whether there's any structure in that population that's unexpected. In this case, the parental samples have been highlighted red, and you can see there's a fairly nice cloud and unstructured population here, so we don't see any unexpected things in this PCA plot. But occasionally you will see, you have some issues with, for instance, unwanted selfings in that population, or an unwanted outcrossing, so a different pollen parent in a population. Those sorts of things can potentially be highlighted by a PCA plot. The next step, well, we often look at the summary of the data, and to see, again, it's somewhat similar to the check F1 step, but it just summarizes the markers, gives you an indication of the counts now of different marker types. So again, the counts of markers which carry in parent 1 a 0 and in parent 2 a 0 or a 1 and so on. And also the percentages of incompatible offspring for each of these different marker classes. The function has an argument for incompatible rates. So if one of these percentages exceeds that, well then, or sorry, an individual carries more than a particular threshold, then it'll be flagged as being potentially incompatible in that population. The following step that we do is marker dosage conversion. Now, marker dosage conversion, again, is to simplify our data set down to a smaller number of marker classes. In a tetraploid, yeah, you can have, for instance, a quadruplex times simplex marker. Well, then we would also convert that to a nullplex simplex marker and so forth. So we try to simplify our markers and to group them. And there's various reasons for that, but the main reason is because of the way polymapR has been programmed. So it's looking for particular marker classes. And so we, by converting our marker dosages, we essentially allow the polymapR machinery to do the analysis. So it's a bit of an artificial step, but it does give us, I think, a bit more of an honest or an informative look at marker densities in our data set. If we look at a quadruplex times simplex marker, that has an awful lot of, if we try to visualize it, I mean, that'll have an awful lot of extra points or extra marks in such a plot. And ultimately, a quadruplex simplex marker only has one interesting segregating allele, the simplex one. So that's the one that we would like to visualize. So having converted marker dosages also allows you to produce more meaningful visualizations of your marker density. Okay, so we get some information from that function mentioning which markers were converted and whether one or two parents were converted, and whether non-segregating markers that were identified were also removed at this stage. There are a number of screening steps that we do. We screen for missing values, and that's both screening for missing values across markers and also across individuals. And again, we set usually a 10% threshold for this. That's up to the user to decide what sort of a threshold to set. And again, based on those thresholds, you can always go back and rerun this step if you find that your threshold was too stringent, or you would like to be more stringent than the default cutoff setting. We can also do that same step for individuals. And again, in this data set, we're not removing any, because although I did simulate some missing values, apparently I didn't simulate enough to make it worthwhile to run this function. But still, these are the standard steps that we run through in the pipeline. Duplicate individuals are not necessarily a problem, but again, they present themselves quite often. So this function, which you've seen in the past, it's looking for correlations, high correlations between marker scores, between individuals' marker scores. So each of these dots represents a pair of individuals in the population. And you can see here 2 pairs have been detected by this function, and they've been merged so that we get the, shall we say, the best possible individual constructed from these 2 duplicate individuals. So there is a merging step which also fills in missing values. So if one of these individuals had a missing value and the other not at a particular locus, then the not missing value would be used. Screening for duplicate markers is essentially a marker binning step. Again, That's when markers are together in a single bin, in a genetic bin as such, so they don't add any extra information. They give exactly the same segregation across the population. So these ones are screened out as well, or at least binned. And this final step is just to pull out the filtered dosage scores from our final screened data object. And if we look at the parental quantities there, we can see again, we now have this simplified set of markers. We're dealing with a triploid population. So the possible marker classes that we have here in the dosage is parent one. The dosages from parent one are here in first position, and that's a diploid. So we're only having values of 0 and 1 in the first position. And in the tetraploid parent, then you can see you can also get duplex scores. Okay, now that's a good time to save our data set. And then we move on in the process to define linkage groups. Now, as I said, parent one is a diploid mother. So although the functions look quite similar, the process is a little bit different than what we've seen in the past with polyploids or with tetraploid data sets. So I'm going to go through this in a little bit more detail. We run the linkage function. There has been a bit of a change in how this function is called from previous versions of the package. In the past, there was particular arguments, target parent and other parent, where the column headers of the dosage matrix had to be provided each time. I decided that was a little bit cumbersome. It's much simpler just to specify which parent you're interested in. So in this new version of linkage, you just specify that in this case, which parent you want to estimate linkage in is in parent one. Again, we have to use the arguments ploidy and ploidy2 to, again, tell the function that this is a diploid times tetraploid cross. And we've told it that there are, in this case, 7 cores available, which is, on my machine, the total number of cores minus 1. We capture the output of this function into something we're calling simplex nullplex parent 1. And then we can visualize that using the orlog plot function. And what you'll see is that again, this is the diploid parent, and that our 2 phases, our coupling and repulsion phase, now are combined in this plot. So usually when you run this in a polyploid, in an autopolyploid, your 2 phases will be separated, and that's because essentially you're using your full population to estimate linkage when it's in coupling phase, but when it's in repulsion phase, only a fraction of the meiosis are informative about recombination between a pair of markers. In a tetraploid, that proportion is 1/3, and that's reflected then in a much lower lot score because your effective population size for the estimate is 1/3 of your full population size. That's not the case in a diploid. In a diploid, whether markers are in coupling or repulsion phase doesn't matter because they're both in the same bivalent. And so each bivalent, each meiosis is informative regardless of the phase between the markers. And that leads to this situation that your 2 phases are, well, they're overlapping in this plot, but because we use LOD scores to do our clustering and to pull apart our phases actually in higher ploidies, it becomes a bit of an issue at a diploid level that we can't separate them anymore. Now, the functions in polymapR are aware of this issue, shall we say? So if you run the cluster SN markers, the basic clustering function of polymapR to do this clustering step, and you tell it that the ploidy is 2, then it will automatically know, okay, well, I'm only expecting here the number of, in this case, the base chromosome number, which for banana is 11. So it'll expect, in this case, 11 clusters, and it'll tell you that as well. So in this visualization here, one of these plots, when we're looking at the number of clusters, you'll see here this dashed red line is at that level 11, and we can see here above a lot of 4 that we find 11 clusters. So we find our 11 chromosomes. We use a different function called phase simplex nullplex diploid. And this function is the function that gives that phase assignment to our markers in our diploid parent. So again, we're just providing it with the output of the linkage function. We provide it also with this cluster list that we have, and that's the output of the previous function. We tell it that, yeah, at a lot of 6, actually I could have even put this at a lot of 4, but at a lot of 6, certainly we find our chromosomes. So this is the lot at which the chromosomes are identified. And again, this LG number is the base chromosome number of the species that we're working in. So in this case, 11. And it happily tells us that we were able to phase all those markers using the coupling information at a lot of 6. If you want to then look at this output, I mean, you can just look at it here. It has 3 columns: the simplex nullplex marker name, which is here, the linkage group number, and the homolog number. And so those homolog numbers in this case will just be 1 and 2s for the 2 phases, and so on. And we can also, yeah, produce a table of those 2 columns. Again, we'll see in this instant, it's a very nice breakdown of markers. So what you want to see here is a fairly balanced number of markers assigned to both phases across the 11 linkage groups, and that is the case in this situation here. Now in the tetraploids, we do it slightly differently. We use the same linkage function. Now we're mentioning that the parent of which we're working in is parent 2. But other than that, it's the same call. I didn't specify multiple cores here, I noticed, but anyway, it's fast enough. If we now plot this recombination frequency versus the lot score, you can see that those 2 phases have separated out as we expect. And that is, as I said, a direct consequence of the fact that in a tetraploid or in a higher ploidy level, we're using far fewer of those meioses to make these estimates of recombination frequency when we're in repulsion phase, because a lot of them just aren't informative, because those 2 homologs are not together and pairing and recombining with each other during meiosis. We use that same function that we had before, cluster SN markers. And it tells us now that 44 clusters are expected. So that's because in this case, the ploidy that we've specified is 4. Now this may be a little bit confusing because previously we were saying ploidy 2 was 4, and here now I'm mentioning ploidy is 4, but this function only takes a single argument for ploidy, and it's the ploidy of the parent that you're actually working in. So in this case, the ploidy is 4. Now, we have all these separate homolog clusters, and we needed another marker type to associate them with each other. And again, this is something that you've seen in the past. We're using the duplex nullplex markers to provide those bridging links. And the bridge homologs. Oh, function will do that. It takes a little bit of time. Okay. Yeah, object O is not found. Try that again. And you can see here, well, if you're willing to count those big blobs, there's 11 of them there. So they're the 11 chromosomal groups again, clustered nicely in that tetraploid parent. Okay. Again, I presented this sort of these sorts of functions in the past as well in more detail. So if I'm going a little bit quickly here, it's deliberate. It's because you've seen this already. Or at least I hope you have. If we look at the breakdown of our, shall we say, occupancy of homologs, so how many markers have been assigned to each of the 4 homologs in the tetraploid parent, well, we can see in one instance, chromosome 3, we have 5 homologs identified, but for the rest, we seem to have 4. So we have to do something for chromosome 3. And that's because we pulled it apart at quite a high lot score. In this instance, we seem to have broken apart. One of our homologs, one of our chromosome groups has been split apart there artificially. So if we first of all visualize that and how these links, how they vary, shall we say, over different range of different lot scores. Well, you can see at the lowest score here, which in this instance is 1.5, a lot of 1.5, we end up with our 4 homologs again. So this clustering at a lower lot score has put those 2 fragmented sub-homologs back together. And we can see here a visualization of the links between them at that level. So if we're happy with that, we just change this argument from false to true. So we modify the linkage group homolog stack, as it's called, and then that's outputted from this function, and we capture it again here. So this is what we've been providing it with, is the original sort of description of our chromosomes and homologs. We now update this by modifying it, and we call it version 2.1. So if we look at version 2.1, we can see here that that issue on chromosome 3 has been solved, that we've merged those 2 homologs together. Now, quite an important step is to check that your numbering of linkage groups is consistent across the parents. We do that by introducing a linkage with a different marker type, in this case, simplex markers. And again, we run it both in parent 1 and in parent 2. So specifying which parent is 1 and 2. But for the rest, the arguments are the same. Okay, so let's run that. Now, I see nothing coming up in the chat, so I'm not sure, are people so far with me, or have they got issues? Or maybe people are just listening, or maybe not listening. It's also possible. Can people still hear me?
Yes. All good.
Okay, so all good. That's good. Thanks, Dave. Okay, so this is run the linkage. Let's see, did I do it for both parents there? Yes. Okay, so what I've done is I've calculated the linkage between the simplex nullplex markers in parent 1 with these cross-parental, these biparental markers. I've done the same thing in parent 2. And then we just use this function to check that the linkage names are consistent. So we go for consensus namings, and we supply what we've just figured out, these linkage estimates between these cross-parental, these biparental markers. Oh, there's a bit of an issue there. Number of levels differ. That's not good. Let me see, why is that happening? Not good at all. Okay. I'm just wondering. Yes, okay. So I needed to run these 2 steps, so I don't think I actually ran these steps. The assignment. Yes, okay. Now, that was a little bit of a minor hiccup. This often happens also when if we're running workshops and somebody says, why is this happening? And I'm looking at the error and saying, I have no idea why that's happening, but it's because they didn't do control and enter on one of these steps. So I just did it myself here. We have to, once we have this information, we have to make assignments of our markers to particular linkage groups and homologs. So essentially, this object here that I missed, this parent 1 simplex assigned, is giving the marker name and giving the assigned linkage group to which it goes, and then also the counts of the number of significant linkages it has to simplex markers on various different linkage groups. So in this case, there were 16 significant linkages, and they were all on homolog 1 on chromosome, or at least linkage group 6, and so on and so forth. So we need that information, because what we're actually going to be doing is we're just going to be tabulating with these cross-parental markers these numbers here. We're going to be tabulating and checking, well, for this marker here, it seems to be going to linkage group 6 in parent 1, and in parent 2, well, I'm not sure if that's also the first marker in the, oh, you get the idea. Yeah, there it is again, the same marker. And in parent 2, it's been assigned to linkage group 1, and so on and so forth. Okay. So if we run the following function, we see the original connection, shall we say, between our numbering of linkage groups in parent 2 and our numbering of linkage groups in parent 1 in these rows. And these are the counts of markers that are in each of those two groups. So it's quite a clean set of assignments. There are some slight off hits, shall we say. So in this case, there's a marker here that doesn't seem to be going with the crowd. But in the most instances, it looks very good. And then it renumbers, in this case, parent 2. That's the one that we want to modify, the linkage group numbering, parent 2. Well, it doesn't matter. We're just by default choosing to renumber the, but again, these are just random numbers. We will see in a short while how they correspond with our chromosome numbers from the reference sequence. But for now, we've got consistent numbering of our linkage groups across parents. And that's, of course, essential if we're going to be integrating our maps. Now, the following steps, now that we have essentially the basic structure of our data set figured out, the following steps then assign all the other marker types to our linkage groups. And these unfortunately take a couple of minutes, and I don't have that time right now to run them. So I mean, I can run these assignment steps, I think, very quickly. But if I run this function, the homolog LG assignment, it's going to take, I think, about 5 minutes or so. And I don't have that time. So I'm going to skip down to line in my script here, line 358. And I'm going to just load back in those marker assignments that would have been calculated here. Okay. And now we can again check that the marker assignments for all the other markers are also consistent across parents. And in this case, it's fully consistent. So that's quite reassuring. And now with our bigger data set, previously we just had the simplex markers. Now we can look at the total assigned markers to each of these different linkage groups. But what's interesting, in fact, is to compare them to their chromosomal assignments based on the reference sequence. So, as I said, the row names are often where the marker names are carried in polymapR. We can split these row names because we can see that the first part of that row name carries chromosome assignment, so we can split it using this this dash symbol, so we can, and then extract. So this slightly complicated little bit of code just extracts out, in this case, the first part of those marker names, and we compare them, we tabulate it against the assigned linkage groups. Yeah. So we can see here, again, a very nice consistency between our linkage groups and these chromosomal assignments. But an issue is starting to emerge here that I suppose you've anticipated, and that's that between, in this case, chromosome 1 and chromosome 4, we're starting to see that our linkage groups are not as they should be, or as we would have expected based on the reference sequence. We can see that again if we do the same arguments instead of parent 1, if we change it to parent 2, the same thing emerges. So more or less a perfect consistency with our physical map, but something strange going on between chromosome 1 and chromosome 4. Okay, now that for now, this issue may be worrying. I mean, I can imagine if this was a data set that you were working on, you're going, oh, there's something wrong here. But as we're going to see in a little bit, there's actually nothing wrong with this at all. I'm going to clear those plots just in case. Now, finishing the linkage analysis means now that we have assigned all our markers to these separate chromosome linkage groups, we then perform a linkage analysis between all those different marker types within those groups. So there's 11 different linkage groups that we need to calculate. That's going to take a bit of time. It takes about 15 minutes, 20 minutes, I think. So again, I'm going to skip that. I'm not going to be twiddling my fingers here for 20 minutes. Instead, I'm going to read in the output of those previous lines at line 410 here in the script file. And then that's object, this combined linkage information from both parents is what we pass to MDS-map. Again, this takes a few minutes, and so you can run it on your computer, but I'm going to skip over it and I'm just going to read it back in. But all those steps are there for you to run either now or at a later time if you want. Okay, now there was a step earlier of having binning markers, so taking our duplicated markers and putting them in bins. And those bins, well, we have them in this screen data 4 object. Actually, in our case, there's only 5 bins. There's only essentially, how would I put this? There's only 5 duplicated markers in our data set. So in this case, this marker here is representing the one underneath it, which was in the exact same bin. So it had the same signature across the population. So the exact same dosage scores in all the individuals. For the sake of completeness, I'll add them back in just to complete the map, but it's only 5 markers. If you want to look at, for instance, the counts of number of markers in each of the linkage groups, you just do an S-apply of the number of row. And in total, there's 4,163 mapped. So I'm going to save that object and I'm going to just read it back in because I called it complete, but I'm just going to overwrite the integrated map list with those extra 5 markers. If we produce some visualizations, we can plot that map. Yeah, you see here, maybe the legend is a little bit off. You can change that legend position to 11.2. Yeah, so this is just, again, well, maybe that wasn't the best choice. A visualization of our linkage map, our integrated linkage map across the parents. So you can see a very high density map, map lengths around 100 centimorgans. Again, this is a fairly nice data set, so would expect the quality of the maps to be quite high here. I'm interested though to check the maps particularly for chromosomes or linkage group 1 and 6, because these were the ones where our physical assignment was not as expected. And you can see in these diagnostic plots, particularly here, again, there's nothing to suggest here that we have an issue with cross-chromosomal linkages or anything like that. This seems to be a very consistent, so low recombination frequencies along the diagonal, meaning that, yeah, our map order is consistent with the underlying recombination estimates. And for the LODs, again, along the diagonal, we see the higher LOD scores as expected. So there's nothing in these visualizations that suggests that we have an issue. What's quite nice, though, to do is to make also a comparison of our genetic and physical map position. So that's, again, something that you saw in the previous speaker's talk, so these so-called Mary maps. And that's quite informative to see that your order of markers is consistent with the reference sequence. Now, what I would like to do first, though, is to, because polymapR uses map lists, so it uses lists in OR. I want to convert that into a data frame. So I just use this small little command to do that. So we're just OR binding each of those 11 linkage groups into a single data frame. So this map now has just got 3 columns with linkage group, marker name, and centimorgan position. I'm going to do the same trick I did before to extract the chromosome number and the base pair position. So that's using these two commands. So I'm going to add on 2 extra columns here. Now you see it there. You could just check to make sure that it's consistent so that we've extracted correctly the marker positions out of the marker names. And again, we can now tabulate what we've seen before. And this is now for all the markers, so for the integrated map. And again, we see that issue between chromosome 1 and 4. Now I'd like to renumber my linkage groups to be consistent with these chromosome numbers. So we just create a conversion lookup table. And we add an extra column. Or sorry, we don't add an extra column here. We overwrite the linkage group with these renumbered linkage group numbers. So again, I'm just calling my linkage group numbers now to be consistent with the chromosome numbers. So it looks like this. And again, we're going to visualize this comparison between the physical and the genetic position. And for that, I want to create some sort of a dot plot. Okay, so this code basically just generates such a plot. I like to have the orientation of these genetic maps to be consistent with the physical maps. And we can see here on linkage groups 1, 4, 6, 7, and 10 that the orientation is in the reverse direction. So we reorient our maps, again, just simply subtracting our map position from the maximum map position on each of those. And then I'll replot it using this. So it just flips them around to get them all lined up nicely. So what we can see here very nicely is this translocation that we had previously found in a mapping study in banana. And again, this is the well-known translocation that occurs between chromosomes 1 and chromosomes 4. So part of this, so on this axis here, on the the y-axis as such, we've got our genetic position. So this is our genetic map position on the y-axis. And you can see here, there's no tension or break between these on the y-axis. It just continues along nicely. But in terms of the x-axis, or the physical position, you can see that that whole segment headed over, and it fused with the part of linkage group 4. And there was a reciprocal translocation in the sense that part of that chromosome also headed back to chromosome 1 and fused with it as well. It's a very nice example. But again, so if I had used the chromosomal numbers for my grouping, this would have caused a big issue with this analysis. The final step is to generate a phased map. The simplest is, I've done a number of tricks now to try to renumber my linkage groups to be consistent with the chromosome numbers. I've also done some tricks to reorient them so that the map orientation is consistent with the reference sequence. So I don't want to, I want to recreate a polymapR map list from my map file. And I just do this, again, it's nothing that complicated. You just, do an lapply and OR basically, and you give it some names, in this case, LG1 to LG11. And now I've got something called integrated map list 2, which looks like a polymapR map list, but it's just been made from that corrected map data frame that I had. Okay. I'll just delete that. And now finally, I just run the phasing function. So this pulls out all that phase information that we've been picking up during the linkage analysis. I think it's also useful to go back to the original coding of your markers. In fact, I recommend it. To do that, we need to redo the marker conversion step and to set an extra argument marker conversion info to be true. The reason is when we're converting markers, sometimes we convert one parent or two parents, and that needs to be known in order to do a correct phase assignment using the original marker scores. So we now have two different versions of our phase map list, one with the original marker scores and one with the converted marker scores. And as I said, the advantage of the converted marker scores for this sort of thing is mainly for visualizations, because we see, shall we say, more clearly amount of the density of our segregating alleles across the parental homologs. Okay. And if we want to visualize that, this is again, I think, using the converted scores. And just for linkage group 1, this is a visualization of the phased map, the phased parental map. And that's what we need for the next step, which is QTL mapping. I still have 20 minutes left. So that's I think I have 20 minutes left. Yes, I do. So that's a good thing. It's about the right time to swap over. I've also provided phenotypes in that workshop folder that was on polyploids.org. If you look also, I mean, at a later stage, I don't know if that link will still be there. These links are also embedded in this HTML. I put them also up in Figshare. So both the marker genotypes and also the phenotypes for the markers to run this are also on Figshare. So you can just click those links embedded within that vignette, and you can also get to the phenotypes there. So in this instance, let's see, I see the row names. I suppose it doesn't matter actually. Oh, okay, that won't work. Hang on, I'll just leave it for now. Yeah, so we have, I would have to use retable here. Okay, it doesn't matter. This is our phenotypes, phenotype file. It's just, in this case, talking about yields. Banana yield, I'm not quite sure if this is kilograms or not. Again, I will admit this is a simulated data set because I had a bit of difficulty to try to simulate a completely non-additive QTL. Well, not difficulty, but I wanted to give some motivation for this workshop. But I would be very interested to see how this also works on real data sets and real examples. The genotypes are, again, the same names that we use in our genotyping files, so that's important. The order doesn't have to be the same, but you just have to have consistent identifiers across the data sets. And in this case, we've a number of blocks in our so a number of replicates, so repeated measurements of yield for our different genotypes. We update the polyQTLer package. Rulan pointed out to me that yes, so I made a last minute change to the update that I had this week, and that hasn't been built into a binary yet. So I'm going to say yes, I want to install it from source. It's pretty much the same, but there's a little bit of a slightly an improvement in version 1.1. Okay, so this is updating polyQTLer for me from the version that you may have had previously installed. Oh. Taking a little bit of time. I guess I didn't need to do this, but this is the joy of running live. Okay, so some of these warnings, don't worry about them. Yeah. This is not a problem. It looks more of a problem than it is, but it's not a problem for now. Okay. Okay, yes, it has now hopefully installed. Yes. Now, I'm going to quickly run through the steps. A little bit of time. So the steps are, again, similar to what we had before. PolyQTLer had to be updated as well to also deal with diploid times tetraploid populations, but that was also okay. In this case, I'm also allowing for multivalence, so I'm mentioning the argument bivalent decoding to be false. and I'm specifying that I want to run it on multiple cores again, just to speed it up a little bit. When I'm supplying the phase map list, I'm using the original scores. Again, it won't make much of it with your genotypes, with the actual markers and the alleles. It's again much more useful, I think, to use the original marker scores for this, but you won't confuse things later on. I'm also going through the step of interpolating those IBDs at a grid of positions at a 1 centimorgan spacing, and that's to speed up the calculations later on for QTL scans. It makes it a lot faster. The splining step does take a little bit of time, but not a huge amount of time. It's always good to visualize these haplotype probabilities using the visualize haplo function. In this case, I'm setting a seed so that what you see will be the same as what I show. Okay, I think this is still going. So I'm just going to sample 9 offspring out of the population and then visualize them in a grid of 3 by 3. So this is the way the package visualizes our haplotype probabilities. And again, you can see clearly the recombination breakpoints, but it's a very clean data set. There's some examples of small double recombinations here, but for the most part, they seem to be very good. But I'm certainly not suggesting that you should just look at a single linkage group. You could look at multiple linkage groups, but this is just an idea. It's good also to count the number of recombinations. Currently, the recombination counts are only being done when it's easy to do it from bivalence. So counting recombinations from multivalence is a little bit more tricky. So only from offspring that are predicted to come from bivalence. Now, this is a very useful step to identify problematic individuals in the population. It's something that I talked about at a previous workshop as well. So again, if we're assuming that there's a single crossover between homologs in a pairing bivalent, then we would expect in this population approximately 33. Just as a sort of a guideline for how many recombinations you expect genome-wide in this triploid population. And you can see in this case, it's a fairly nice distribution, but in a real data set, oftentimes you see a number of individuals which might have 200 or 300 predicted recombinations. So extreme outliers in this distribution, and they would be ones to look at more closely and essentially probably kick out because of some quality issues. We also often calculate the genotypic information criterion, or the GIC. I'm just going to show it here for 2 linkage groups. And again, while here you can see in the diploid parent, actually the GIC for both of those homologs completely overlaps. So we cannot distinguish any difference in information content of our two phases or 2 haplotypes in the diploid parent. But that's as expected. In the tetraploid parent, we can see here, for instance, a nice example of a single marker at the end, at the top of homolog 4 leads to an increase in the information content of that particular homolog, whereas the other ones decay, having no marker coverage. So this is a representation of the amount of information we have. Again, it's very informative to look at that in your population. I want to visualize my phenotypes, and for that I'm using the lattice package, so you may need to install this, but I'm just doing a scatter plot between the 3 different blocks, the 3 replicates, and to see is there consistency in my yield values across those 3 different blocks. So it's here you can see it's extremely consistent. So calculating blues for this should be no issue at all. So we run the blues function. Again, this is to estimate these best linear unbiased estimates. In this case, we're treating the genotype as fixed. We just want the estimates of the genotype of the individuals in this population. We essentially get an average for these phenotypes across our three different blocks. And if we add that into this visualization, then you can see again that the blues are very consistent with these underlying measurements in these three different blocks. So instead of using the full structure of the data set with the experimental blocks, which is possible, I'm just going to provide these blues because it just means that the subsequent steps for QTL mapping are a lot faster. Now, the original, so the former version of the QTL or for QTL scan didn't have additive effects. I just have to mention that, yeah, we want to run a permutation test. So we want to estimate significance thresholds. And then I'm using the plot QTL function, which also has been updated since the last version, to visualize the output of that QTL scan. So here we see, again, across our 11 linkage groups, we find a major effect QTL on linkage group 1. There's some sort of a minor QTL on linkage group 2. And for the rest, there's nothing particularly large effect in terms of QTLs on the other linkage groups. Now, what is new is the ability to also specify that we want to also include interactions between alleles in our model. So in this case, we're running the complete model, which uses these genotype probabilities in this association. It takes a slightly longer than the previous step, but I still think we have a bit of time for this. Okay, so running that again took a little bit of time, but we can plot the output again using this plot QTL function. And as I mentioned in the vignette, this function now has been updated so that you can not only provide it with the outputs of this QTL scan, but you can also provide a list of different outputs and it'll plot them together. And if you do this in this particular format, it'll rescale them so that these thresholds will overlap completely. So here we see, we see the outputs of, on the first case, the additive effects model, and the second case, this full model, compared side by side. And what's interesting is in the case of these full model, this complete model, we detect an extra QTL that was not picked up by the additive effects model. So there's a QTL here which is curious in the sense we missed it in our original scan, but we now find it with our scan that also allows for interactions between our alleles. Now, in the final steps, once you have identified a QTL peak, you may want to explore what are the effects of the alleles underlying that QTL. So we use the explore QTL function. The output of this is a visualization of the BIC. So essentially sort of a likelihood measure which compares the likelihood of different models, taking into account the complexity of these different models, these QTL models. And each of these numbers corresponds to a different QTL model being tested at that particular peak. In this case, we have a number of QTL models that are quite likely, similarly likely, but the most likely in terms of the BIC seems to be having an allele, the segregating allele or functional allele at that QTL carried by homologs 1 and homologs 4. And we can see that again with this visualization. These are the effects of these different homologs plotted across the whole of that chromosome. So around the peak position, you see indeed that from homolog 1 and also from homolog 4, that's consistent with what we're seeing in terms of the effects, in terms of increasing the yield in this population. For linkage group 10, we do something slightly different. We actually capture the output of this function. So this function explore QTL silently or invisibly returns some information, but if you don't capture it, well, you won't see it. It won't be returned. But if you assign the output of this function to something, you'll see now that you get a list with various different things in it. In this case, for linkage group 10, this was this QTL that popped up only using the complete model. So this is the QTL that's missing if you just use an additive effects model. And so if we visualize our QTL effects just using the ordinary call that we've used in the past, again, the QTL is at the top of this linkage group. It's somewhere around here. And yeah, you could try to connect and make some sense of which of these parental alleles is contributing to this trait. But of course, this is a little bit misleading because these homologs or these alleles on their own do not describe the variation that we see at this locus particularly. Instead, what you need to do is you need to specify that allelic interactions is true. So you need to specify that you're interested not in the shall we say the haplotype means for each of these particular haplotypes, but you're interested in seeing the mean effect of combinations of particular alleles. So at the moment, this is the visualization that you'll get. We included the possibility of double reduction when we're estimating these genotype probabilities. So that's why you get these extra classes 3, 3, 4, 4, and 5, 5, and 6, 6. In this case, there's no colors in these because the probabilities of these is 0, and you can actually see that if you pull out the genotype means. This is essentially what's being visualized. So these are the means of each of these different parental allele combinations, but in the ones which carry a double reduction, it's missing because the probability of those classes in this instance, in this data set, was 0. And that's because I didn't simulate any double reduction when I was generating the genotypes. But for the rest, we see here, for instance, the combination of alleles 3 and 6 in parent 2 leads to a sharp decrease in the yield. So there's something, this interaction and if offspring have both of these alleles together, that will lead to a particularly poor phenotype. And then a more favorable combination of alleles, for instance, would be the combination of 3 and 4 together, or 5 and 6. Okay, now I see I've come to the end of my, oh no, almost the end of this. The last step is to build this, let's see, what have I got here? Yeah, okay. Let's skip that. I think I've said that already. So the last step in this today's workshop, and we'll be finishing now in a couple of minutes, is to build a genome-wide QTL model. So building a sort of a multi-QTL model, and that's again, I showed this previously using the check cofactors function in the past. So this goes through all the possible combinations of cofactors that could be fitted to try to increase the precision of estimates for other QTLs. So try to build as big a possible QTL model to include as many as possible significant QTLs. Because of course, if we account for a big effect QTL, we may detect other smaller effect QTLs as afterwards. It does take a little bit of time because there's quite a few different combinations that are done. And just to mention, this has also been updated because now I'm mentioning that I want to test for the full model as well. By doing that, I'm also saying that a cofactor can be fitted not just as an additive effect cofactor, but as shall we say, using as the cofactor rather than just the haplotype probabilities. So I'm not sure if that makes a huge amount of sense, but it because you're also allowing for the fact that the cofactor is either additive effects or additive plus interaction effects. Yeah, it runs about twice as long as before. So it takes about 5 minutes for this to complete. So I think this is good because I think we are through today's workshop for polymapR and polyQTL, just about in time.